Oh, there we go. Perfect. Can you hear me now? All right. Well, we're off to a great start. Thank you for everyone letting me know. All right. I know it's working now. The problem was just the microphone has a little bit of a mute button on top. Perfect. Okay, so we're good to go now. All right, so just to recap what I was talking about while I was muted, Jeff isn't able to come today. Jeff is working on something really, really, really cool. He's been working on it for like the last 2 weeks. It's something, yay, perfect. Everyone can hear me now. Awesome. Let me know where you're coming in from in the world in the chat. I always love to see where everyone is coming in from. But what we're planning on doing today is still covering the in vivo CAR T space. But we're going to be doing something a little bit different. Since Jeff isn't able to join, he's working on something really cool. He's working off of like 3 hours of sleep over the last 7 days. So we're just going to let him take a nap. What I'd actually like to offer you guys today, if you like, once we kind of go through the initial section where we just introduce the in vivo CAR T space, is if you'd like, you can come on the show for 3 to 5 minutes. And so I've put a link in the description of this episode that should be there now. And if you'd like to come on, you're more than welcome to come on the show. Perfect. All right. I'm seeing we got someone from Seattle. Excellent. We're what the plan we're planning on. Oh, we got someone from Florida. Perfect. This is awesome. So the plan is today, if you would like to be part of the show, if you'd like to come on and provide your perspective with regards to in vivo CAR T, if it's just hype or if it's just a bunch of, or if it's actually got something that's legs, if you'd like to talk a little bit about ex vivo, feel free to click that link in the description of the episode, and we should be able to bring people in onto the show for a couple minutes each, just to kind of provide your perspective on it. Excellent. All right. And we've got Yun Chang, a PhD student at UT Southwestern. Awesome. And working on non-viral gene delivery. Perfect. So you let me know if if I mess up anything, Yun Chang. And maybe if you'd like to come on a little bit later, we can talk about your research. Excellent. Okay. So just with regards to what all the hype is around in vivo. The really interesting thing about this modality is that it promises to be something that's going to be ideally a lot more logistics proof. One of the major selling points for in vivo, if we just back up for a second, is the fact that you're only using a viral vector or an LNP or some combination of the two, and you are going to be directly inserting into a person's body this instruction set that's going to equip the native T cells with the instructions that they need to recognize a cancer or recognize some sort of bad thing if it ends up being an autoimmune disease or whatnot. And so the idea here is you're going to have something that's a lot more effective from a logistics standpoint, because instead of taking, well, I guess I can just bring up the graphic now. So let's, if I add this to stage and do this, okay. So this is actually a graphic I made 6 or 7 months ago. It might be out of date, so you guys can let me know if something on this is wrong now. Maybe there's something that's come out in the last couple months. But overall, there's some pretty interesting advantages for ex vivo. And I think the most important one to look at here would be the cost and logistics part of it. So when it comes to ex vivo, in a standard sense, the problem has been you've got to take the cells out of the patient. You've then got to take them over to some sort of GMP facility. You've got to introduce the genetic material like the CAR that's going to allow it to recognize its cancer. You've got to then grow up these cells into an army of millions. You've got to make sure they're properly activated. You've got to make sure everything's hunky-dory. Then you've got to transport them back from the facility to the patient, and you've got to make sure that everything's all QC'd up and ready to go. It's a really complex logistical burden. It's not undoable, but it's certainly very difficult. So by contrast, the benefit, the promise would be for something like in vivo. You can come along and you're going to be able to just give a person one single injection, and that's going to be the end of it. You'll be good to go. So as a result, the promise here is it could be something that's quite disruptive with regards to the rest of the industry. And so everyone's very much interested in it, because if you can now do this at a much less logistical burden, ideally it's going to cost less. The other big advantage of in vivo right now as well is the fact that you don't need to do a lymphodepletion step, ideally. So again, this was something I didn't fully appreciate until far too late in my career, just because I didn't start with a cell therapy background. When you go through a standard cell therapy, you've got to undergo a lymphodepletion set. You've basically got to knock out the native immune system entirely so that it doesn't reject the millions of new cells that you're going to be infusing into the body. So you've got to basically blast someone's entire immune system, clear everything out, and then reintroduce with the new cells that you're bringing in, a couple million cells, and that's going to repopulate your immune system. That's a problem because it's obviously pretty damaging. It's not ideal. It can cause a lot of different health defects. If you end up getting sick while you're lymphodepleted, you could get very, very sick and even die. And so with something like in vivo, if you can come along and remove the lymphodepletion set, you're going to be in really good shape. So again, let's just kind of look through the different components of this. So again, with regards to that treatment timeline as well, right? With a ex vivo CAR T cell therapy, you're going to have to, again, go through that huge logistical process that's very costly and just takes a long time. And so if you've got someone who's on death's door with cancer, even if you now qualify for CAR T, you're still a couple weeks away from being able to receive it into the patient. And so the concern is the patient could get that much worse, and then the treatment isn't going to be effective, and they end up passing away. With in vivo, the promise, right, is that you would just be able to make a huge bank of this injectable ahead of time. You don't have to worry about some sort of delay that way. And so as soon as the person qualifies, give them an injection and they're good to go. And the other benefit that people are talking about with regards to in vivo as well would just obviously be the scalability thing. And so when you're thinking about making hundreds of thousands of personalized doses and scaling that to the masses, it's a very, very difficult problem. Obviously, not as much of a difficulty as like there's people who are working on it. It's something that can be done, but it's obviously going to be very difficult. If you guys have any questions or anything at this point, feel free to jump in. We can talk about it. We're just doing a little bit of an introduction right now, and then we can open up the floor a little bit more to discussion, because Jeff currently is operating off of like 3 hours of sleep in the last 7 days. He's been building something really, really cool that we're going to be able to share with you soon with regards to new founders, developing and getting in contact with the correct investment firms. Super cool. Stay tuned for that. But in the meantime, if you have any questions, put them in the chat. Or also, if you would like to come on the show for 3 to 5 minutes to talk about your perspective of in vivo versus ex vivo, there should be a link in the description if you're watching on LinkedIn for the video there to come on the show. So I'd love to see some of your beautiful faces up here next to mine, since Jeff isn't available today. Okay, so not only do we have some benefits with regards to in vivo, with regards to it being a bit more easy to scale versus ex vivo, but there are obviously some challenges, and it's not all sunshine and roses. And I think this is probably where we want to be thinking a bit more about where in vivo is going to fit into the wider conversation. One of the big problems with in vivo at this point, and this is something I kind of saw over and over again when I was doing some research on it to prepare for this show, is that at this point, it does have an unproven track record. There hasn't really been any clinical data released. Correct me if I'm wrong, but there hasn't really been a whole lot released from the clinical perspective to showcase this is something that works really, really well. Maybe there's stuff behind closed doors that has driven all of this investment. But we just don't know about it yet. But that has been the challenge that myself and Jeff have seen as well, is this is still something that's early. There's not a whole lot of clinical data for it. And the other challenge, obviously, will just be as well the efficacy part. How well does this work? When you have an ex vivo cell therapy where you are controlling all of the cells in a controlled environment, you're making sure that you can look at every single last one of them, basically, to make sure that they've got the correct set of instructions. And then you're infusing that quality of cell into a human, into the patient. That's different from what's happening with in vivo. With in vivo, you're just sticking in the genetic information and putting it into the bloodstream and hoping that it gets to all of the different T cells and that it integrates correctly and that you get a significant enough number of those T cells that are going to uptake the information for, like, the chimeric antigen receptor, for example, and hope that it works. But we haven't seen a whole lot of data yet, just with regards to how that actually translates to clinical results. Again, the promise and the potential is there. If we can make something where we have an in vivo CAR T happening, where you just have to give it the instructions and your immune system takes care of the rest, that would be great. Okay, Rachel is saying, ESO Biotech published initial data in Lancet from 4 patients treated with in vivo CAR T therapy. Okay, interesting. So I'll have to go look at that. But still, 4 patients, fairly early days, right? That can be me and 3 friends who it's good data, but we definitely want to see more than just 4 people. But this is good. Thank you, Rachel, for that. So maybe we can bring that up a little bit later. I might have to look that up on the side and see if we can look at what the results are for that. But that's the challenge, right? Is across a very diverse patient population that is our world, are we going to be able to very consistently, just by injecting in the LNP or the viral vector, get a significant enough result that it replaces the need to take any of your cells out of your body, make sure they're fully ready to go, and then go back. Okay, and Stuart Gibb is saying ESO is also speaking at CAR T London in the new year. Expect a well-attended talk. Yes, maybe I can make it to that one. Stuart, if you don't mind me asking while you're here, what time is that conference? Like, is it CAR TCR? Anyway, I'll move on, and then we can come back to that. But yeah, that'll be very interesting to see, because yes, they've been leaders in the field. Okay. The other thing that we need to be thinking about with regards to in vivo, not only do we not have the ability to see yet whether it's going to be completely effective, the challenge is we literally aren't able to really take a sample of the cells to see how well they're doing in real time. And kind of what I mean by that, right, is like you don't have that ability for the cells aren't readily available for analysis. Like, you could take a blood sample, obviously, and then do a bunch of different things to it. But the challenge is going to be, compared to ex vivo, where you're doing very, very hefty QC, you're making sure that the cells are of a certain quality before they go into the patient. It's a lot more difficult to do that after. To actually look at the cells now that they're circulating all over the body. You can try to take a blood sample. It might not be completely representative of what's going on. And so the challenge here is just, are you going to be able to really monitor it in a way that showcases that it's effective? That might be something that's coming. I haven't seen that yet. Okay, perfect. Yes, so it is CAR TCR in February. Interesting. All right, I might be able to go to that one. We shall see. And I would definitely want to attend that talk with ESO Biotech. Okay, on top of that, the other challenge with in vivo is it's relatively new. And with the fact being that it is relatively new, we don't know yet if there are just going to be new risks associated with in vivo CAR T. The challenge again, right, is when you do a gene editing event, or you do some sort of viral vector going into like lentivirus that's going into cells that are ex vivo, and you're putting something on that's pretty potent, right? You're talking about a chimeric antigen receptor. We're talking about like a killing tool that we're now putting onto very specific cells. when we're in the controlled environment of a cell culture plate or a T flask or a G-Rex or whatnot, and we're now looking at those cells and we're able to assess how, and obviously we're just looking at the T cells. We're making sure it's just the T cells that are inside of this reactor. We're only editing those cells and then growing them up to a specific standard of quality and then reinfusing just that into the patient. We're dealing with something that's completely different when we're just willy-nilly adding in a full-on chimeric antigen receptor and anything else and knockouts and edits into the body. It's just a completely unregulated environment. It's the Wild West. It's dark. It's complex. Everything's changing everywhere. And so are there brand new risks that we may not have even considered yet that aren't in the cards yet because we haven't really come across it? Are there brand new risks with adding in something as powerful as a chimeric antigen receptor, and it could end up on all sorts of different cells, not necessarily just T cells? Obviously, that can be solved somewhat with targeting, but there's always going to be some amount of off-target effects, and the question is going to be, when you're introducing something that potent, is it going to be a problem? Okay. So we're just about done here. The one other thing that I just wanted to quickly talk about here at the end, right, just with regards to off-target effects, that really is one of the big advantages of ex vivo so far. Ex vivo has been able to control everything in this to the one side. You're really able to look at the cells and analyze the quality of all of them before you put them into the patient. Such is not the case as much once you actually just are hoping that you're making all of these different CAR Ts in vivo, and it's much more difficult to assess the quality. So that would be one leg up for ex vivo as well. Yeah. Okay. So again, folks, originally Jeff was going to be able to come. If you're just joining now, Jeff was going to be kind of taking a bit more of the devil's advocate. Now he's going to play the middle a little bit more. But Jeff is operating off of like 3 hours of sleep over the past 5 days, 6 days. He's been head down building something really, really cool with regards to connecting early stage companies looking for investment with investors who actually care about the science that you're doing. And the early results that he's shown me, it's absolutely insane. But yeah, he's dog tired and he's kind of been frantically working on it, and he's made something really cool. So stay tuned for that in the coming days. But as a result, what we're going to be doing that's a little bit different today, now that I've just about finished up this intro between in vivo versus ex vivo, and we're just kind of looking now at what the future will be, I would love to extend the invitation to you guys. I know we've got some questions here in the chat, and so we can get to those as well. But if you'd like to actually come on the show for a couple minutes, there should be a link in the description if you're watching on LinkedIn. And you can click on that. It should take you through to StreamYard, which is like the platform I use to run the live stream. And if you'd like to come on and chat about it for a couple minutes, I would love to have you, because me just chatting by myself sometimes can get a little bit monotonous. But before we do, so if you'd like to, feel free to click that link and I can bring you on the show for a couple minutes. And even if you want to talk about like the research that you're doing with regards to in vivo or ex vivo, love to do that as well. We can keep it a little bit more open-ended. It doesn't just have to be your perspective on whether you think in vivo is hype or hope. Okay. Okay. Yes. Oh, good question. Yes. Yun Gong. Have we talked about our view on the therapeutic window? How big? Yes. So this is the interesting thing too. Ex vivo has benefited from the fact, and I had a chance to talk with someone who had CAR T at CAR TCR in Boston a couple months ago. And she got it, 7 or 8 years ago, and she still has CAR T cells, now that she's cancer-free, floating around inside. Oh, okay. Thank you for letting me know, Stuart. Let me fix that. So the link apparently isn't there. So it's not so it should be in the description of the video, but maybe I put it in the wrong spot. Okay. Well, regardless, I will get that link just a second and get that over to you. Just a moment here. Copy invite link. Maybe I'll just put it in the comments here, folks, and you'll be able to see it. One moment here. All right, I'm going to add it in as a comment. And hopefully you should see it there. Okay. So it's a StreamYard link. I've put it in the comments. So if you got, for those of you who have been very kindly commenting, you should see it there down below. Okay. So if we back up here for just a moment. Yeah. So you were talking, Yun Gong, about the therapeutic window. Ex Vivo has showcased that in certain conditions, right, you will still have protection years and years and years later. So if cancer ever shows up again, you've got T cells right there, boom, good to go. The challenge will be with regards to in vivo, can it actually do the same thing, or is this only going to be a temporary result? You only get some T cells sufficiently activated with your with your chimeric antigen receptor. That ends up going off and helping with cancer for a little bit, but it doesn't give you a lasting result. That remains to be seen. Maybe if you've got more to add on there, Yun Gong, I'd love to hear. But that would be one of the challenges, right? With the different folks that I've spoken with so far, if you're using something that like an LNP and you're just delivering like an mRNA, it's going to be very minimal in terms of the amount of time it's going to take. However, if you've got a very viable gene editing strategy that is able to create a progeny of CAR T cells that last a long time, that would be interesting. However, again, the challenge being, is it actually going? Are you able to actually generate edits at sufficient numbers in the wild west that is the human body and everything that's going on there? Very good. Okay, and then Cesar, I cannot open that link, so I'm not sure what it is. So if you're able to describe what it is, that'd be great. Okay, so I'm pretty much done at this point. There's a couple other things that I want to talk about throughout the show with regards to in vivo versus ex vivo. But in terms of my introduction, I'm pretty much done. If you guys can see the link in the comments there for StreamYard, and you'd like to come on and talk about this for a couple minutes, that would be great. And we'll see if this kind of thing is something that you guys like. We might end up doing it again in the future. I am experimenting today. Cool. All right. Just while we're waiting. Oh, Yun Chang. Good question here. I don't think it's about the duration of effects. I guess what do you mean by that, Yun Chang? Like what do you mean by the duration of effect? If you'd like to come on the show, you let me know, and you can just click that StreamYard link. Okay. All right. So I'm sorry for whatever reason. Sometimes StreamYard is just like, we don't know who this person is, so we can't share their face. So I'm sorry, anonymous LinkedIn user. So Novartis T-Charge platform, 3 days transduces cells ex vivo and lets them expand in vivo. Isn't that a safer approach than in vivo CAR T? Now, this is a really good point. I tend to agree. Especially if you have a population, right? I believe of stem T cells that are going to actually be the long-term memory of it. I like that a little bit more, right? It allows it to be a little bit faster. And this is kind of also the other thing that we should consider in this conversation as well with regards to in vivo versus ex vivo CAR T. With ex vivo, we're often kind of comparing it to the status quo of right now, where it's like, yes, you've got to take the cells out of the patient. You've got to take them over to a different facility. You've got to transform them. You've got to grow them up. You've got to ensure that everything's super good. Then you've got to bring them back. You've got to make sure all your QC tests are done, and then you can put them into the patient. However, that is kind of the standard way it's been done in the past. But what if point of care systems end up crushing, right? Where the patient lives like in a bed right next to their cells as they're getting made. And it's basically just a vein to vein situation where the cells actually never leave the hospital and they never leave the hospital room. And so with regards to the Novartis T-Charge platform, 3 days, if you can master some sort of point of care system that does that right in the hospital room, that might end up being the nice compromise in between. But again, right, the huge benefit to in vivo, if it does play out the way it should, is again, you're able to take the cells and you don't have to touch the cells at all. You can stock up a huge bank of the therapeutic ahead of time, get all the QC done, and as soon as the person's ready to go, boom, there's no delay, and you can go do it right away, hopefully with no lymphodepletion as well. So that's the question. Okay. Here's the question. Okay. Here, Yun Chang. Correction. The therapeutic window is defined as the range of plasma concentrations in which a drug achieves the desired effect with minimal toxicity. Referenced. Yeah. Very good. Okay. The other thing that might be interesting to consider here, and again, folks, Jeff isn't able to join today. He's operating off of like 3 hours of sleep over the last 6 days. And so we're just trying a bit of a new format today, where if you'd like to come on the show, there should be a comment in the comments that I linked to a StreamYard platform. And if you'd like to come on the show, let me know, and we can chat for a couple minutes about what you think about the whole in vivo versus ex vivo thing. I think the other thing that will be interesting to look at as well would be the broader potential therapeutic reach. So obviously, like for people who don't know, I'm a gene therapy guy. I got my start doing upstream and downstream AAV production in a little lab in Guelph, Ontario at the university. And so obviously I love gene therapy. I think it's awesome. There's super cool things that have already happened in the field. There's more things that are happening that just make it really, really promising. Obviously, over the last couple of years, it's been a bit more difficult, but ultimately I still believe in the long-term viability. And so with something like in vivo, right, you see something that's so elegant where it's either just one vector or a couple different vectors that are working together to produce an effect in vivo, and the patient gets to live an incredible life. Most of the CAR T applications so far, right, have been limited to cancer, a little bit inside of autoimmune, but mostly inside of cancer. With something like in vivo CAR T, does it allow you to kind of go after new indications that ex vivo CAR T can just never hit because it's not worth it, right? The reason that we've gone after ex vivo, we've gone after cancer with like cancer cell therapies. Yeah, shout out to Guelph. Sweet, thanks, Leah. The really interesting thing about in vivo CAR T is since you don't have the same potential worries about lymphodepletion, you might not have to worry as much about cytokine release syndrome, that sort of thing. It means that you can go after indications that aren't necessarily quote unquote as serious, right? Someone's not going to die next week the way it is with cancer. And so therefore the risk profile isn't as much of an appetite. There isn't as much of a risk profile appetite there. And so with something like in vivo, if you don't require the same, if there isn't the same risk profile there from the CRS side and from the lymphodepletion side, does that mean you can go after other diseases that could still benefit from a specific type of T cell that's recognizing something bad and destroying it, but there isn't the same burden of risk? So that could be interesting. But again, we just don't know at this point. Okay, Leah, you had another question here. Could you tell us more about clinical trials or companies focusing on in vivo CAR T right now? Yes, so I can. So one in particular that I posted about earlier this week, just a second here, is Azalea Therapeutics. I hope I'm pronouncing that correctly. And Azalea Therapeutics is doing something that I think is super cool. If I can bring up their website for a second here. They are, I'm going to see if I can just share my screen. So this is kind of what kicked it off. They have a really, okay, let me see here if I can share. All right, can I share my screen? Okay, so this is a recent, so this is a company that literally, well, it didn't launch this week, but it launched its Series A this week. So it just closed a Series A for like $82 million. It's crazy. It spun out of the Doudna lab. So obviously the one of the ladies who helped discover CRISPR-Cas9 in the first place. And they are doing something really cool where they are combining together two different different vectors. So they have their envelope delivery vehicles. And then I saw like in a press release, I'm not seeing it here. But they're also using AAV. So they're using these EDVs, these envelope delivery vehicles, as well as AAV to deliver different components into the body to produce the effect of making T cells super, super effective inside of the body. And so they're really focusing on the fact that we're able to get to a very, very, very precise cell type and do a very, very specific and precise gene edit. And so therefore it minimizes kind of those off-target effects and some of the risks that we may not have even considered at this point. And so they're thinking about that, but it still somewhat remains to be seen. Oh, I've been sharing it the whole time. I am sorry. Here we go. Can I do this? Okay. So this is Azalea Therapeutics. Highly recommend checking them out. Again, Jennifer Doudna is the co-founder. So, the company is in good company. And yeah, they have some sort of EDVs that are designed to specifically locate T cells within the patient and deliver the genome engineering cargo. And so, yeah, they're also using AAV, which I got my start in when I started in gene therapy many years ago now. So it's pretty cool. Okay, I will stop sharing that. So yes, to your question, Leah, that's one of them that you can take a look at. Some of the other big news in the space that kind of prompted this whole conversation has been the fact that like AbbVie spent like $2.1 billion to acquire Capstan doing in vivo CAR T. Gilead spent $350 million to acquire Interius back in August. Bristol Myers Squibb spent $1.5 billion to acquire Orbital just last month. And then obviously the other one that people have been talking about in the chat as well is ESO Biotech. So ESO Biotech was acquired by AstraZeneca. So there's been loads of activity just from like the billions of dollars flowing into this space. And so the question on everybody's mind is just like, what the heck? Why? Of all the things, like it seems to be somewhat of a new modality that's not fully proven yet. What kind of data are people seeing behind the scenes that's just making their eyes open up and they're throwing gobs of money at this? So that remains to be seen. Yeah. Okay. So perfect. Thank you for everyone coming in here and just giving Leah a boatload of resources. This is awesome. Thank you. It makes my job a little bit easier and I don't have to go back and try to respond to the comments and everything. Okay. So again, I would love it if someone would like to try. And I know I'm kind of just foisting it on you, but if you'd like to come on and talk about this for a couple minutes, I put a comment in the comments where you can actually click on a StreamYard link. You can come into the studio here and we can chat about in vivo or ex vivo CAR T for a couple minutes. But I also know all of you are a bunch of shy scientists. So if you don't want to do that, that's fine. Okay. But so again, that invitation is open. First come, first serve. The link is in the comments. The other thing, just with regards that might be interesting to think about with regards to in vivo, right, is just the fact that you also have this opportunity to combine it with gene editing. So with gene editing, not that you couldn't do it in ex vivo, but the benefit can be compared to something like the COVID vaccines, right? It was delivering mRNA for the spike protein, and that's something that's going to be temporarily expressed inside of your cells, and then it's gone. And so if you're looking for a long-term therapeutic effect above and beyond just activating the immune system once, you're going to want something that sticks around a little bit longer. And so with gene editing, now you're in a position to actually make those long-term edits inside of the body. And it's a double-edged sword for sure. You could have long-term persistence, but if you get it in the wrong spot, or if it's not precise in exactly how you do it, you could get a lot of long-term off-target effects that could be very detrimental to the patient. But what's really interesting about Azalea Therapeutics that launched their Series A with like $82 million. Oh, there's Stuart. All right, let's give this a shot. Perfect. All right. So yes, just to finish my thought, the thought here being, right, with gene editing, if you do it right, you do have this ability to deliver a therapeutic effect for a very long time. Without lymphodepletion, without the same risk of CRS, that sort of thing. All right, Stuart, you are on the air. Thanks for coming in.
I figure I jump in and be your first guinea pig here. Great to see you, Ben. So anyone who's doing this, you have to mute your main screen first, otherwise you get this echo effect. So a quick point. I mean, I think one, it's the future. It's same as we look at allo kind of CAR T. I think for first and foremost, I think, auto CAR T is going to change for the future. I think, we're already looking at different lymphodepletion regimens. It's going to affect that. So that's kind of one piece. I think industry driver, we all know we need to reduce costs, so, reach more people, etc. But I think, note of caution is, this is, again, relatively new technology. So, new approach. We're probably not going to see it as an approved therapy 7-10 years, right? That's a kind of average. But we're going to see headwinds, right? Any new technology, whatever it is, we're going to see some kind of headwinds. We don't know what that is, right? So is it, you say we don't need lymphodepletion, but what about the inpatient costs, right? Or the inpatient, caveats to what we're going to have to look out for. Dosing is one thing. One thing we really don't talk about for autologous CAR T is the possibility of redosing, right? So that's, is that going to be possible? And then kind of, long-term follow-up, what's the caveats to that? So I think we'll see some kind of headwinds. I don't think, having a headwind to a technology is a bad thing necessarily. But, what's that going to look like? And, I think we have to be careful as an industry. I think we're suffering a little bit from it right now, is that overpromise, right? For, what is the efficacy of the treatment itself, how many people we can reach, the costs, etc. So that's just kind of, in the grand topic of, autologous ex vivo CAR T, in vivo CAR T. That's my concerns right now. We've been through this several times, right? Even like the current, set of cell therapies prior to that, we had some, misgivings back in the day for, like, I used to work on CNTF, for example, right? That, killed a lot of people in clinical trials for affecting liver. So I think, we just have to be a little bit careful of the perspective. It is a bit of a gold rush right now. Everybody's going after it. So, we'll see what happens. But I just wanted to jump in since we're all shy scientists, as Ben said, and that's my point.
Appreciate it. Thanks, Stuart. Yeah. So I guess like just one question there, right? So what would you say, right, would be from your perspective, what would be the major risk that we should be looking at for in vivo? What would you say has kind of got you thinking, all right, this is going to be the biggest challenge?
Not to sound like Donald Rumsfeld back in the day, the unknown knowns. I mean, it's the body's biological reaction to this approach, right? So, how are we going to target those particular cells? How are we going to get it to the right area? What are the off-target effects, right? And I think it's those off-target, or the immune reaction to this approach. How are we going to do that? Once we've got protocols set up around that, again, with the lymphodepletion piece, right? If CRS kicks in or ICANS kicks in and it becomes a major problem, there are now protocols to deal with that.
Yeah.
But that's taken time. And that is not the same way of dealing with things from institution to institution. And we, the data's in there with clinical trials. It's just nobody really wants to talk about it.
Yeah. Yeah. And that's the other thing too, right? Like with regards to you saying like the unknown knowns, right? how do you properly assess if you are sticking a gene editor inside, and you have long-term expression? Like, how are you properly able to assess what's causing what? Or at some point, right, like 4 or 5 years down the road, oh, actually, it turns out you've been building a boatload of CAR T receptors in your ankle bones, and now it's super. Now you got a lot of like joint pain or something, right?
I mean, we always talk about the kill switches, right?
Yeah.
When we've gone, and so to explain kill switches, and then you're bordering into IP territory, right? What does that look like? You know, how have we made this safe? I'm sure, like, the regulatory authorities are like, you know, not happy about it. They have to deal with a new set of challenges that's going to make things complex. So I think that's going to be part of the discussion moving forward. How quickly that accelerates, obviously, is out of our control, but is going to be an impediment to getting to market.
Yeah, I agree. Yeah, and it'll be interesting to see where that shakes out. Yeah, with regards to kill switches, this is one thing that some people have said as well. Like, this would be an advantage of in vivo over ex vivo, because we actually have something built into each and every one of these where we just whoop, it's gone.
Yep.
And 6 months from now, once you're cancer free, we can go in and administer the kill switch or something, which is much harder to do if it's a cell therapy.
Exactly.
Very good. Perfect. Stuart, thank you for coming on. This is awesome. Thanks so much. All right. Bye now. All right, folks. It's as easy as that. Thanks again, Stuart, for coming on. If anyone else has some questions, we've still got a little bit of time here, and we can talk about it. In particular, Yun Chang, if you're still here, I'd love to hear about what research you're doing over at UT Southwestern, but no pressure whatsoever. Okay, Leah, good question here. Speaking of headwinds, what kinds of pushbacks have people seen from regulatory agencies specific to in vivo CAR T? So this is a good question, and folks, feel free to correct me if I'm wrong, but one of the challenges with this right now is it's still early. The FDA doesn't know what it doesn't know. And so I imagine that these different therapeutic developers are having conversations with the FDA with regards to what should be happening and how to work with all of this. But the FDA ultimately won't have a lot more information than we do right now. And so they will be cautiously looking at everything that's going on. They'll be looking for those early risk factors and then developing guidelines around it. But you see something similar with even gene therapies, right? As it relates to regulatory agencies, like early, like in like, I think it's just fairly recently that the FDA has actually started to talk about with regards to like AbbVie, for example. So forgive me, guys, this is a little bit of a nerd session, right? When you're making a gene therapy, right, you have a protein case and you've got your therapeutic inside. The goal is to build, make billions of these different protein cases with your therapeutic gene inside. That then goes into all of the cells that need it, administers a therapeutic effect. You're golden. Problem is, biology is complex and biology is hard. And so what you end up having with AAV, right, is you have the protein case, but it might be empty and there's no gene therapy inside of it. You might have the protein case and it's just got a chunk of random DNA inside of it, or just a portion of your therapeutic payload, and so it's useless. It's junk. And then you actually have your full your protein case with the full therapeutic inside that's actually going to administer the therapeutic effect. And so at first, people just didn't really think this was that big of a problem. It's like, oh yeah, you got some fulls, you got some empties, that's fine. And you just kind of want to make sure you've got a decently okay full empty ratio, but it's not something you really need to look at. But it started becoming a much bigger problem. It was causing all sorts of immunogenicity problems. A couple of kids died in a trial where the quality of the vector wasn't good, and at the high dose that they were administering, it was just far too much for the kids. And so the FDA starts to come in, right, with some guidance around, you actually should be characterizing how many of your fulls you have, how many of your empties you have. And then even over the last couple of years, in a company that I worked at called Nanomosaic a couple of years ago, well, last year, they were developing a device that allowed you to actually characterize your fulls, your partials, and your empties, because it's not just the full empty ratio anymore. You want to be looking at the fulls and the partials, because the partials are often useless as well, and so you don't want to be making those. So all of that to say, when we come back to your question, Leah, about the headwinds for in vivo CAR T from the regulatory side, that might still be coming, right? They're going to be reacting to kind of like bad things that happen as they occur, and then making rules for everybody else, being like, don't do that because it'll end up with something bad. So that's I think kind of where we with regards to regulatory, it's very much a wait and see. We aren't seeing a whole lot yet, but ideally we should into the future. Okay, just a comment from Elia here. I think ex vivo is becoming more powerful and close to succeeding in clinical trials. Well, ex vivo actually already is, Elia. That's the awesome thing. So there's a lady that I know, and she had like stage 4 leukemia and took a CAR T that was available, and she was completely cancer free and still is. So yeah, ex vivo is awesome. It's continuing to succeed, and we'll see what happens into the future with regards to it. The challenge right now is just the deliverability of it. It's somewhat difficult to make a dose. I know a lot of different people are working on technologies to fix that, but that has been the challenge, and that's where in vivo therapeutics are kind of coming in and being like, hey, we can fix a lot of the problems that ex vivo have, but they also will have some of their own challenges. Okay, Alexander with a good question here. Can you talk about the current state of non-viral delivery for in vivo, specifically the challenges? Good question, Alexander. I am by no means an expert on this, but we did have a chance to speak with a gentleman from MaxCyte a couple months ago. I think it was back in August. If you go, let me just show you here. I'll put it. Actually, what I'll do is I'll just put a link to it in the chat. But we had a conversation with a gentleman over at MaxCyte a couple months ago, and we talked about this very thing. They're obviously really big on non-viral strategies, and we talked through that. All right, it's somewhat. It's somewhat. Oh, here we go. Right. It's somewhat incorrectly labeled as the case for lipid nanoparticles. MaxCyte does a whole lot more than just lipid nanoparticles. I'll put. Oh, just a second. I put it in the wrong spot. Here, I'll put it in the comments here. Oops, there we go. Stop that. Okay. Okay, there's the other event. So yeah, coming back to your question, just with regards to non-viral. So they have come a long way. The huge benefit of non-viral delivery is the fact that you can, to some extent, make you can make the individual unit far more consistent compared to some viral vectors in the past, right? I just went through talking about how you have fulls, partials, empties. And that's just because biology is super complex, and you're making them inside of these, inside of HEC293 cells inside of a lab, and the cells are floating all over the place, and not all of them are reacting the exact same way. And so it's difficult to get the consistency super high with non-viral delivery systems. You can, like specifically LNPs, you can get around that problem where you're just delivering, where you're able to get the individual unit far more consistent at scale. And so that'll be super relevant when it comes to in vivo. It's like, okay, we know that we're putting a far more consistent product inside of the patient, and so ideally that builds the case. Therefore, the results that we've seen, where we're saying we can very precisely target certain populations of cells, and we can very precisely do gene edits, since the individual unit is so much more consistent, we can reliably say that is happening at a very high rate inside of the patient as well. So that would be probably one of the things that I'd say with regards to that, Alexander. It is interesting as well, just to even see some of the new types of nanoparticles that are coming out, not just lipid, but other materials. And there can be all sorts of interesting things there. The other thing that can be interesting as well is with regards to non-viral, but it's not going to be interesting. You're not going to be able to do it for in vivo, but even just like electroporation. Obviously, you can't do electroporation inside of the body. That zapping an entire person with electricity isn't going to work super well. But there might be some interesting approaches, right, where you could actually take someone's blood, you run it outside of their body, you electroporate the cells that are inside of that, put them back inside of the person, and you minimize maybe some of the challenges there. But that remains to be seen. But lots of really cool technology happening with regards to that. Okay, let's see here. Let's see if there's anything else I just wanted to talk about briefly with regards to in vivo versus ex vivo. So I guess maybe as we're in the last 10 minutes here or whatnot, the interesting things to consider as well would just kind of be like cost. Cost of goods for in vivo could be a lot lower, just from the fact that you don't have the same logistical burden. But then, like for right now, with regards to ex vivo, you're typically looking at somewhere in the range of like 300 grand to 500 grand a dose all in. Not a lot of people going to be able to pay for that. So the potential benefits of in vivo being able to come along and say, hey, we can make these batches in huge amounts, store them, and then just pull out a little bit at the right time. It could have a pretty big benefit with regards to cost, but that remains to be seen, because obviously we'll have to see if the cost of manufacturing for those very consistent in vivo vectors that you're making are going to be lower than the standard right now. The other thing to consider there too would be ex vivo cell therapy developers and technology developers aren't just sitting on their hands. Yes, right now it's 300 to 500 grand, and that has limited uptake even with approved products. And so obviously there's a $1 billion challenge here. If you can make a very Morgan is saying here, ESO Biotech announced 13,000 COGS per patient. Whoa. Okay. Yeah, that's quite a bit lower. It'll be interesting to see what kind of markup they put on that. But again, right, it'll be in the context. Thank you for that, Morgan. I'm just going to leave that up for a little bit since we're talking about cost. That's going to be really important for people to consider. If let's say that they still want their cake and they eat it too, and so the COGS are $13K. They're going to sell this for $100,000 to make their money back from all the clinical trials and development, or even $150,000. You're now coming in at like 2 to 3 times less than the current standard. That's big, and it's going to mean that 100 to 150K is now kind of more in reach for people who want to do like a crowdfunding initiative for their aunt who's got some sort of cancer. It's much more realistic than $300,000 to $500,000. Yeah, that's a good point. Wow. Yeah, 13K is crazy. And it'll be interesting, even with Azalea Therapeutics that I mentioned earlier. They're using AAV as well. There should be some interesting things coming out in the next couple of years with regards to really reducing the cost for making high quality AAV as well. I get to talk in some of those circles, and it's pretty nifty. So long term, I'm quite optimistic and bullish about the ability of these individual components, especially the vectors and the LNPs, coming down significantly in cost. And they've got to, if we want to get to those really large patient populations that are going to help a lot of people, but ultimately will mean that the market and the money to be made will be bigger. It's way easier. It's you're going to make more money if you can, care for 10 million people at $100,000 a dose than if it's only 50,000 people at $1 million a dose. Or I hope the math checks out, but yeah. Okay, that sounds good. Stuart Gibbs. Oh, and talking about Jason Foster. Well, there's another name that I love. Jason Foster said the cost is 50% inpatient care, hence cheaper off the bat if all good. However, let's see if there are any reactions in patient needs. So yeah, this is crazy with regards to, if you're thinking about kind of the standard for cancer right now, this is kind of outside of in vivo versus ex vivo altogether. The really challenging thing, right, is if you've got like a small molecule, it's a pill that you just take with some water. I'm going to take some water because I'm parched. And all that a person has to do if they've got like some sort of ailment, like Tylenol, right? Let's use Tylenol as an example. You don't have to go to a hospital to take Tylenol. You don't have to be monitored while you're taking Tylenol. You can just take Tylenol, you screw off the crap, you pop a pill in your mouth, take it with some water, and you're done. That's it. No further burden required. And even with some different like cancer therapeutics, right? It's a small molecule. You can take it home. You just drink it with some water and you're done. You're not assessing the patient the full time. With cell and gene therapies, that is not the case. Once you administer that sucker into somebody, you gotta be watching them like a hawk, and you've got to be administering it a very specific way. And so therefore that's just contributing. It's not the actual cost of the therapeutic itself. It's just everything that's required around actually administering it to the patient. And so that's going to be another area where if someone can come along, and maybe this is something that like the robotics industry will just be able to really crush and help out with. And it's just going to be, can I make the inpatient costs and the costs around the actual administration of these therapies still be regulatory compliant, but dramatically reduce? It'll be interesting to see. And that is currently a challenge right now that people kind of don't talk about. We often just kind of talk about the cost of the therapy itself, but then all the costs surrounding that and monitoring the patient and making sure everything's okay and that they don't get CRS and nothing else goes wrong. Maybe as the risk profile for some of these gene therapies and cell therapies comes down, that won't be the case as much anymore. And that helps reduce the cost, but that is ultimately something we're going to have to look at. And if someone has a good idea with regards to that, that's probably $1 billion idea that someone would pay you a lot of money for. So think about that. How could you better care for and monitor patients in and around receiving a cell therapy or a gene therapy? That makes it more conversation just about the cost of the therapy itself and not everything surrounding it. Okay. We're just about finished here. Here's a question from Cesar. I cannot see myself, so I'll just read it here. What in vivo CAR T can allow with off the shelf is benefiting economy of scale. Yep, agreed. One batch could ideally treat more than one patient, not mentioning that also underdeveloped countries, which can't afford the ex vivo manipulation protocol, could also benefit from it. Nevertheless, in vivo needs clinical robust data. Yeah. If we could kind of just sum up the whole perspective of in vivo right now. First of all, Cesar, and I hope I'm saying your name correctly. I apologize if I'm not. And it's a quote. It's a quote I came across when I was doing some research. It's this: the variability of in vivo. Or sorry, where is it here? Oh yeah, the viability of in vivo CAR T's efficacy is unproven, but strongly anticipated. So it's currently unproven, but strongly anticipated, right? And so that's really going to be the key. If this does work, it will mean dramatically wider access. And that's obviously what we're hoping for. But at this point, we are in a wait and see pattern. We've got to see if this thing's actually going to work and see if it justifies the billions and billions of dollars that have been poured into it. Obviously, we should all hope that it does succeed. Patients are waiting. But ultimately, we are in a wait and see pattern, and that's probably just about where we'll have to leave it. Excellent. Well, thank you everyone for coming today. Again, if you haven't heard already, Jeff wasn't able to make it today because he is operating off of very little sleep over the past 5 days because he has gone down this crazy rabbit hole of making a really cool digital tool that helps early stage biotech founders get connected with the right investors. More on that coming soon. It's super cool. He showed me some of the early results already. Blew my mind. If you'd like to learn more about that, hit him up or ask me after and I can connect you. But really, really interesting stuff. And we'll be releasing more about that soon. But that's about all I can say publicly for now. Oh, okay, perfect. Caesar, perfect. All right, I will do that. Thank you. Like Caesar salad. Excellent. All right, thanks so much, everyone. Appreciate you coming, and we will see you next time. Cheers.